sequenced, and you want to know what is the proportion of different source environments to this sample, right, that contribute to the sample. So yes, for that we created decOM, and decOM outputs something similar to this, a bar plot that tells you, or a CSV file that tells you this is the contribution of this source and this and the other. And the second method I'm going to be talking about today, which was something that we thought would like come up intuitively, is once I know my sample is contaminated and how contaminated it is, I might want to get rid of that contamination. Can we come up with methods to remove that contamination from the FASTA files, right, at the read level? For that, we developed a k-mer-broom, which removes contaminant reads. So basically gets rid of this part, right, like the skin and soil contamination that I guess you guys are not interested in when you are working on ancient oral data. So the first method, again, it's called decOM, and it's a similarity-based microbial source tracking method of ancient oral samples that uses k-mer-based methods. And again, to keep everyone on the same track, microbial source tracking is a process to quantify, or a method to quantify the proportion of different microbial environments that are also called sources in a target microbial community that is also called a sink. So you have your sample of interest, you want to know how contaminated it is, this is going to be called a sink, and you also have an intuition of what are the environments that contribute to the composition of that sink, and these environments are going to be called sources. So for that, what we did is that we gathered a collection of 360 samples coming from all around the world, including contaminants and non-contaminants. So basically, the composition of those samples are, the 360 samples is roughly 32% ancient oral samples, 23% modern oral samples, 23% roughly skin samples, and the rest sediment and soil. So we gathered this collection of samples, metagenomic samples from everywhere in the world, from different sequencing centers, and we built a k-mer matrix with a tool called k-mer-tricks. So k-mer-tricks is a tool that was developed by one of my colleagues, and I also deeply recommend it in case you want to represent your data, so a set of metagenomes, or actually genomes also, in the form of a matrix. So what is a k-mer matrix is nothing but this two-dimensional representation of the data, where the rows represent the distinct k-mers and the columns represent your samples, right? And you have ones whenever the k-mer is present in the sample, and zeros when it's absent. So it's a binary k-mer matrix, what I'm going to be talking about mostly when I talk about decOM. I also wanted to mention that the very, very useful resource that you guys have collaborated for, and that has been like widely used for the community, Ancient Metagenome Directory, was the source I used to collect these ancient oral samples, but also I used the microbiome database, the human metagenome database, and the Magnify resource from the EBI, to gather this collection. So how does decOM work? So decOM, it's a comparison between a vector, which is going to be your sink, your sample of interest, against a matrix of sources, which is composed by other metagenomes that include contaminants and non-contaminants. So to give you like a more graphical view of the method, you can think of it as a problem of balls that fall into bins, right? So what's going to happen is that we have our k-mer matrix of sources, and then decOM is going to represent your FASTA file, your sample of interest, as a vector, binary vector, where each of the rows corresponds to a k-mer, a distinct k-mer. So if you see this sink here, your sample of interest, the darker gray is going to represent a k-mer that is present in the sink. And in this case, we can see that it's present in the sink, and it's also present in 3 of the sources. And these sources are labeled as soil sources. So what I'm going to do is that I'm going to put 3 balls in the bin of soil, right? I'm going to continue with the next k-mer. The next k-mer is absent in your sink, so I'm not going to add anything there. Then I'm going to add, to continue with the following k-mer. The following k-mer is present there, but it's also present in skin source, in 2 of the soil sources, and in 1 of the ancient oral sources. So I'm going to add 1, 2, and 1 respectively to each of the bins. And I'm going to do that until I finish all of the k-mers. And then I'm going to come up with proportions, right? So you see here that the largest contribution comes from soil, and because we evaluated our method as a multi-class classification problem, then we're going to give a hard label. We're going to say the predicted label by our method is soil because that's the highest contribution. And then we're going to evaluate the performance using that. But roughly what you need to understand is it can be equated to, or it's equivalent to a problem of balls falling into bins, and it's simply counting the occurrences and and then estimating the proportions. So for that, we evaluate the performance with different metrics, such as accuracy, F1 score, precision, and recall in different experimental setups. First one was a leave one out experiment, where we simply take the matrix and then take out one of the column vectors and compare it against the rest, and then put it back, put the column vector back, and take another one and compare it against the rest, and that's called simply leave one out. For that, you can see that the performance of decOM is considerably better compared to the rest. We also evaluated it on an external validation set of ancient oral samples. Again, the recall is higher for decOM than for the rest. We additionally performed a cross-validation experiment to account for biases that might come from simply like being able to predict the class of a sample because it was part of the same bio project. So this cross-validation, I think, has something similar to the leave one out, but specifically splitting it in five folds of the k-mer matrix, and four of them would be left as sources, and the fifth fold, right, the fifth group would be taken out as sinks. And then we performed different experiments and evaluated the metrics, and you can see that for accuracy, F1 score, precision, and recall, decOM not only performs better in each of the folds, but on average as well. And then finally, we performed a synthetic dataset experiment where we evaluated the method on a mock microbial community of ancient oral samples, and we see that decOM is able to estimate a higher proportion of oral content than source tracker and FIST, which were the competing methods we were testing our method against. The distribution of the method, so it was presented last year as a workshop paper in Learning Meaningful Representations of Life, which was a workshop for NeurIPS as a machine learning event. 2022 is currently under review in microbiome. All code is public in a GitHub repo, and it's also available as a conda package. I wanted to mention something that might be of interest for you, which is, okay, how does it compare to FIST and MetaSource Tracker, but in not so many numbers? I just want some words to give, so have an understanding of it. So decOM is alignment free, whereas FIST and Source Tracker need a reference database. The input, sort of the matrix of sources, that the k-mer matrix of sources in decOM is what would be equivalent in FIST and Source Tracker to an O2 table, which some of you might have heard of, right? Like an operational taxonomic unit table that come with diverse discussions that I believe we have had in the last two SPAAMtish regarding on how to build a table, how to normalize it, and all so on and so forth. The three methods are adaptable for other types of metagenomic data. decOM was evaluated and created specifically for ancient DNA. FIST has not been so widely used for ancient DNA as far as I know, whereas Source Tracker has been widely used in many environments, including ancient DNA. There's no parameter tuning needed. In decOM, all you need to know is how many resources you need, and you can allocate to perform the job, right? Like how many threads and how much space memory you can allocate for your job. Whereas for FIST and Source Tracker, several other studies have mentioned that you actually do need parameter tuning, optimizing parameter selection for each of the problems you're going to solve. decOM is a deterministic method. As I mentioned, it's just counting, and FIST and Source Tracker are probabilistic. They're Bayesian methods, and so they might give you very different results if you run them with the same parameters and the same input data. The difference the variation on the output depends on the method and the input data, of course. And then I planned the live demo. Let's see if it works. So basically, I just wanted to show you the GitHub repo, which is this one here, where we briefly explain what the method does and a bit longer what it does. We mentioned the installation requirements. You have to only like clone the repo, create a conda environment, and activate it and make sure the tool is on your path. And then, as I said, we have pre-built a k-mer matrix for you in the case you want to test if your ancient oral samples are contaminated, which is the k-mer matrix I described just before. So you just download it and you unzip it. And then you can perform a test with the test data that is there. Let me see if I can open that. Whoop. So decom runs for one single sink as just this parameter decom. You indicate the name of your sink. You indicate the path to your sources folder, right? The sources being this k-mer matrix that you downloaded and untarred. The key file, it's simply a file where you indicate the name of your sink and the location, right, of the FASTA file. Nothing more complicated than that. And then, as I said, how much memory and how many threads you want to allocate. And it starts running, right? So while this is running, I wanted also to show you a bit some of the other stuff you can do with decom. So again, if you have several sinks, you can also test several sinks, which is this that I mentioned here. Then you should have as an output, right, this CSV file that tells you simply the counts to each of the bins and the proportions, estimated proportions, the running time, and the predicted label, which would be this decom max. You also have a PDF with this bar plot that I showed you and an HTML file in case you wanted like a more interactive version, and then the vector that is created, which is the sink mentioned, binary vector. These are more things about the usage. If you have paired or single end reads, one sink or several sink, and then we've added additional features such as ancient or allowed, because having discussions with collaborators, they mentioned they might be interested in having a k-mer matrix of sources without ancient oral samples because people are afraid that they are very contaminated, that they shouldn't be used as sources, etc. So there's like this other matrix of sources that we have pre-built for you in case you might need it. There's a much general version of the method, which is called MST, decom MST. Here I explain how you can create your own input matrix of sources, what would be equivalent to the O2 table. For that, you do need certain amount of resources, but it's doable. And once you create your input matrix of sources, then you can run decom on your own without the pre-built k-mer matrix that we have done. Of course, that would take more time. And finally, the last feature that we added was a leave one out feature in which you can, in case it's something that you're in, if you have your set of sources and you want to test one source against the other and do that for every one of them, then you can perform this experiment. And so just to show you what's happened with decom. So basically, yes, just says decom started with certain version. What are the parameters? All of this is stored in a log file for you to know what were the parameters run, what was decom doing. Hopefully also help debugging and help me out in case you find any issues. And you can, of course, post them on the GitHub repo, ask questions. And then finally, the vector for the sink vector is created. Then, because we represent that as a matrix product of some estimation is computed. And then finally, the assessment for the sample is done. And then you can see this is the folder I mentioned before, right? decom output with the different plots, etc. So that's all for decom, I think. And then briefly, I wanted also to talk about, a much recent method that we have developed is called akmerBroom. It does ancient DNA decontamination, bloom filters on k-mer sets. So basically, as I said, the question we had before was, once we know a sample is contaminated, whether because you use source tracker or FIST or decom or whatever, then is it possible to remove this contamination on the read level? And we thought, yes, we think we can do it using Bloom filters, right? Get rid of this part. So what is a Bloom filter? A Bloom filter is a probabilistic data structure. It's basically a way to represent the data that allows you to query fastly in a memory efficient way if an element is part of a set. That's all, right? It gives you false positives, but no false negatives. It's a very powerful data structure that was created a long time ago in the 70s, I think, and we thought this is a good structure to use in our problem. So what we do is that working on what we had already done for decOM, we said, let's select a subset of k-mers from this large matrix, of which I haven't talked actually, like I haven't given you the numbers, but it's a very large matrix what you come up with when you're working with k-mers. So what you do is you subselect a set of k-mers that are most likely oral, at least in our let's say arbitrary definition, right? So these are the k-mers that are present in any of the ancient oral samples or present in any of the modern oral samples and absent in all of the skin and absent in all of the soil samples. So we take that and we say these are like 1.5 billion k-mers, and we build a Bloom filter on that. And we say, okay, this is kind of like a reliable set of k-mers. And this is roughly the pipeline. So if you have understood what I've said until now, this is pretty much what I've described. We have a collection of data sets that include contaminants and non-contaminants. We build a Bloom filter for that. And then you guys have your ancient oral samples that you've sequenced with a lot of effort, and then you have already your FASTA files, and you want to decontaminate them. So what akmerBroom does is that it k-merizes, so it splits into chunks of k-mers, your reads, and it's going to perform a first lookup against this Bloom filter, right? Then if a read, has two consecutive k-mers, meaning two consecutive k-mers that are part of the Bloom filter, this read is going to be called anchor read. These anchor reads are going to then be part of a Python set, right? And then we're going to perform a second lookup on those reads, on the same input reads. After the second lookup, then we're going to have proportions, something called an anchor proportion, which is basically the proportion of k-mers that were part of the second Python set. And we're going to define, or we're going to set a threshold and say, if a read has over 50% or more of its k-mers present in this set, we're going to say it's ancient, it's oral, keep it. If it's not, leave it out, right? So that's pretty much the cleanup process. Simply a 2-round lookup. That's it. Okay, so we evaluated our method akmerBroom on 3 synthetic data sets, on 2 real oral data sets, and compared it with deconseq and recentrifuge, which were the methods as far as I understand, all perform the same task of doing contamination removal at the read level, not at like the O2 level and so on. And then we see that the performance of akmerBroom on synthetic samples is quite good. So basically, sensitivity it's above 89%, and sensitivity being the percentage of ancient oral reads that were successfully retained, and specificity is also higher than 94% for 3 of the synthetic cases. Specificity being the percentage of non-oral reads that were successfully removed. We also evaluated on real data, which was the only case for which we could compare against competing methods, because competing methods required reference databases or negative controls. You have to understand that here it's much more difficult. That's actually one of the biggest problems to do benchmarking on real data in this field, at least as far as my experience is concerned, because we don't have like we don't know what is the true contamination content of a sample, right? We're just trying to find ways to assess that. So yes, we evaluated performance to real data sets, and we see that the percentage of reads retained by competing methods such as deconseq and recentrifuge is quite low. So it does remove, let's say deconseq for this data set removes a lot of the has a high oral content when we evaluated with source tracker using an auto table, the same auto table that we had built on this like a large collection of samples that I described before. So the oral content is high, but also you remove pretty much everything. So that doesn't really like perform the task. Recentrifuge performs slightly better, but then our method has a good read retainment and still preserves a good oral content above 75%. This method was accepted as a proceeding tracks paper for RICOM-SIC in this year, in a month. And it was also selected as a manuscript for iScience. All code is public in a GitHub repo. We invite you to use it. And just to give you again an overview of the three methods, so akmerBroom is alignment-free, k-mer-based methods have this like potential, whereas the other two need a reference database. With akmerBroom, the user might create their own Bloom filter. So we have also like sort of opened this window for you guys to say, I'm not actually interested in ancient oral samples. What I work is in other stuff. So I want to come up with a set of k-mers and do that type of cleanup. This can be done. In decOM-SIC, the user might create their own index, and in Recentrifuge, the user needs to provide their own blanks. There's no parameter optimization needed in akmerBroom. We have done it already, which is basically like 3 parameters that we can discuss further if you guys wanted. And whereas for decOM-SIC and Recentrifuge, there's a bunch of sort of decisions that you have to make that I'm sure that if you have tried out any of the methods, you have come up with this, right? Like, okay, BWA, which is the algorithm that it's used for decOM-SIC needs certain parameters to be run. How do I run it? Should I optimize this? Is there a benchmark already, etc.? There's also an option to have a remove or retain database, another decision you have to make. Centrifuge, which is this taxonomic classifier used by Recentrifuge to perform the cleanup, also has a bunch of parameters to optimize. So yeah. The 3 of them are adaptable for other types of metagenomic data, and akmerBroom was evaluated and created specifically for ancient oral DNA. The others were not. And here's, well, not really a demo, but I just wanted to show you again the repo. It takes longer, so I'm not going to show the demo, but basically, here's the repo, akmerBroom. We explain roughly what the method does. You build a Bloom filter or you download it if you guys want to use the one we have pre-built for you. Then for a set of input reads, if you have 2 consecutive k-mers in a read, then it goes to a Python set, and then you do the second lookup, and then you get your cleaned sample. These are the 2 options, right? If you have the ancient Bloom filter provided, or if you want to create your own Bloom filter. This is the input data that you need, either the Bloom filter or the k-mers, and your sample of interest. And then the output is an annotated FASTQ file with sequence ID, read length, if there was a consecutive match found, so if the read was considered to be an anchor read, and an anchor proportion, so that you know this read was removed because the anchor proportion is below 50%, right? There's also test data, and that's it. And just to end, I want to say, that I believe k-mer-based methods have a huge potential for contamination removal and contamination assessment of ancient metagenomes. And I find them particularly interesting because they are able to make use of all the ancient DNA studies and all the genomic, like all this effort that the people have done throughout the years, because they can represent this as in many different like data structures, right? And so you can like leverage all the effort done by people throughout the years to come up and analyze your experiment using that information, and not just like out of the blue as if you had just started. And that's it. Thank you so much.
Thank you, Camila. So I guess if there are any questions, now is the time. And maybe, yeah, Tarjan.
Oh, yeah, thank you. Hi, Camila. Thanks a lot for the talk. I think this is a really useful tool that I've been trying out already a little bit. So I only have one particular question. regarding the decOM. So I assume that the more data that you have from different sources when you build the matrix, so the more sensitive the detection in your sinks is, right? But then, of course, the problem would be the available resources that you have to build the matrix. Is there maybe a minimum data input from specific sources, or at least like relative proportions of those that you would recommend using? Or maybe if you stumbled upon that or assessed it in any sense?
Yes. Okay, so that's a super cool question. So we built a k-mer matrix. I can maybe show you the numbers. Here, right? We built a large k-mer matrix that had like something like 9 billion k-mers, only with these 360 samples. That was too much to do anything, right? Even though we rely on Dask, which is a library that does parallelization, and it's really cool, it's too much. So what we did was we enforced one partition only. So we sub-selected 40 million k-mers only, right? It's few, but then we tested our performance, and that we prove in our paper with seven partitions, sorry. And then we saw that the performance wasn't actually improving a lot. So we were using more resources without actually improving the performance for our task, which is a multi-class classification problem. But for your problem, if you're telling me, oh, actually, is it true that I'm gonna get more or better results if I add more columns, right? If I have more samples? I personally haven't tested that. I think that's like, to answer that properly, we have to like actually perform the experiment. I would just to answer the question simply, I would just enforce KM tricks to build 1, 2. So there's like, we can discuss offline if you want, or also it's very well explained on the repo of KM tricks. You can simply select part of the matrix, not the whole thing, because specifically for this purpose, which is like a much, like, I don't know, like a rougher proportion, it's what you need. You need to know if it's contaminated or not. You don't need specifics about the k-mers. Then I would simply just enforce 1, 2, 3 partitions, depending on how much space and time you have. Yeah.
All right. Yeah, thanks a lot. This is actually really useful. I'll try it out, and maybe I can write you on email or over Slack if I stumble across the problems. But yeah, thanks a lot.
Sure. No problem.
Yeah, Anna.
Hi, thanks very much for your talk. That was really interesting. I had a question also about decOM. So I've been using Source Tracker in the past, and I believe that one has like a downsampling step. And I was just wondering if there's like any form of normalization in decOM, and if not, if that would influence the output at all.
And there is no normalization. There is just like a simple counting and proportion estimation. Part of the work that I have been having to do now regarding the reviews has to do with sort of tuning the contamination estimation. Instead of just like focusing on the hard labels and predictions, it would be an experiment like worth doing. I'm guessing things change, but in principle, I'm not sure if like normalization or something more like a correction on how you estimate the proportions, or if you give a weight to the counts. That's something you can say. Like, if there's, let's say, I'm much more interested on you keeping the ancient oral, or like counting for the ancient oral stuff, and then not taking into account the other, then you could weigh the counts and change the proportion. No, I haven't done any type of normalization. I guess that would be something worth comparing. But again, the matrices are different, right? Like, what decOM works on, it's a binary k-mer matrix, and the other table, which is also a matrix that is used by Vista Source Tracker, is a no2 table, right? With counts. So it's not binary also.
Right. Yeah, that makes sense. Thanks very much.
Maria?
Hi, thank you very much for the talk, Camila. It's great. This is a really basic question, and I think the answer is going to be basically yes, but I just wanted to ask for confirmation. So you were saying that for decOM, you can make your own k-mer matrix of sources. So presumably that means that you could adapt it to a different set of metagenomic environments like coprolites or something like that, right? And then could you also transfer that information into the akmerBroom so that you could remove, for example, if something looked like oral or skin and you're like, no, I just want fecal microbiome, then yeah.
Yeah. No, it's, I mean, it's what I said. That's great. I thought I wasn't super clear. Yes, exactly. You can do that with your own source environments, with your own question of research, and then extract those k-mers from the matrix. So right, like these ones here, if you know already the ones that you're interested in, and then use that as an input to a k-mer-broom. Exactly, that's what we did. Of course, there are technicalities to it, because that was like sort of the path I took, which is not like entirely sort of standardized, but yeah, like filtering the matrix and this. And if you guys wanted wanted a hand on that, like just ping me and yeah, I can do that.
That's really great that you've put that flexibility in. It's very useful. Thank you.
Well, nice to hear.
Irina?
Hey, yes, thanks for this talk. This is really useful. And I actually have a question about the k-mer-broom aspect of it, because I know we've talked a lot about this in other SPAAMtish discussions. So I wanted to ask, what actually is the advantage of taking out contamination at the read level over taking it out in a taxonomic table, say? Is there something that is, I guess, does it benefit us more than just doing it at the taxonomic level?
Let me think. I wouldn't know. I would have to think about it if there's an advantage in particular. I mean, I guess that's what the path we took, because we are more used to like k-mer methods than like taxonomy-based methods, right? But I wouldn't know specifically, like if there's some sort of metric that we could use to say, oh, actually, you see, you have a better read retainment and you keep a better oral content if you use it on k-mers instead. Like if you use akmerBroom as opposed to decontam or these other methods that are using like removing contamination from the O2 table. The thing is, I guess also because we have been experimenting on using O2 tables, and there's like this huge discussion which I still struggle to understand, and we've been having on how to process these O2 tables and so on. Then I guess we kind of like look to the other side, but that's a great question. I should keep it in mind. Yeah.
So then, sort of related to that, have you ever done comparisons of the tables of like taxonomic profiling of your FASTQ files before and after you've done the contaminant removal with akmerBroom and seen whether or not it actually does reduce the amount of contamination that you pick up?
Okay, maybe yes, by using source tracker. Maybe that would be the answer, but give me a second. Here, for instance, right, we use source tracker, but like not I wouldn't know specifically what things changed on the table, right? Just by running source tracker before and after removing the contamination with akmerBroom on the 3 synthetic data sets. then you can see that actually the oral content, which is this blue and red bar, it's larger right after removing the contamination. So that's something we did. And we also did it for the real data sets, but we had to scale it for like how much for read retainment, right? How much of the content was preserved. Yeah. That maybe answers.
Thanks.
Mhm.
Yes, and I guess Nico has a comment on the latest question. He says that taxonomic tables have a large proportion of unclassified reads, while k-mer methods are not affected by the incompleteness of the database for annotation. So maybe that's one of the advantage, perhaps. I think Sterling.
Yeah, I had a question. I was just wondering, and this kind of builds off Irina's question. Do y'all have any like extraction blanks when you're processing these samples? So then, because I don't see it as included in one of your source environments, and whether or not, that's additional like source of contamination, whether, and like to go back to her question, then you can compare like if those rates are being identified as contaminants in those sources, then you do the taxonomic profiling to see what those reads are actually like being classified as, and like are they like a more oral or maybe a laboratory contaminant? Have you like done anything or considered anything like that?
Yeah. Yeah, to be fair, the strength of akmerBroom is not cross-contamination. That's something we discussed, like we wrote on the discussion. We really should like try to come up with more experiments and ways to account for that. In that sense, I do have to say that centrifuge, it's kind of like focused on that type of contamination, but we do not include the blanks on the O2 table. So you're right.
Okay.
Okay, maybe I have a question. So why do you use the Bloom filters and the Python set in a k-mer-broom?
Okay, so as I said, the Bloom filter is a very efficient and fast data structure. Been implemented in many languages, quite old, and so we started off with that, right? We started with one lookup of the Bloom filter. The Bloom filter has a positive rate that can be controlled, and one of the ways we found we implemented to control for that false positive rate was adding a second round of lookups. It was supposed to be a Bloom filter too, but due to like sort of time restrictions, because we had to like send the paper, it was faster and easier to implement a Python set, to use a Python set. But that's something we want to do in the future, so that the second round of lookups is done as a Bloom filter.
Thanks.
Mm-hmm.
Any other question?
I have another question then, if no one else does. So for the oral sources that you used from the supplemental table, I see that you have both calculus and teeth. And from one of the papers from Tina's group that's several years old now, they showed that the preservation of oral bacteria in teeth is hugely variable. So when you include something like that as a source, you have a lot of both endogenous DNA in some samples and a lot of non-endogenous DNA in other samples because they're mostly degraded. So how are you selecting what you're actually using as your ancient sources before you kind of have assessed whether or not they're well preserved or degraded?
Yeah, so to be fair, the selection was quite rough, was like an informatician type of selection of the samples, right? So there's this database that has a collection of ancient oral samples, which of the samples were isolated from humans, which ones were oral, that's it. There's no further, like, more deeply grained selection of that. That's the metadata that we used. That can for sure be improved. That's why, like, if you guys have a much reliable set of sources, and you can like come up with your own k-mer matrix of sources, running it in this like decOM MSD implementation, then that's probably gonna give better results. So yeah, that's most likely a source of bias in our results.
Oh, thanks.
Actually, I also have another questions, always regarding the k-mer matrix. So like if this tool becomes always very popular, like everybody uses it, year pass. I mean, you showed the map in the beginning from which you took all the sources. I mean, there are a few areas of the world that are not represented, for example, which I think it's due to the fact that maybe we don't have actual sources of soil, skin, or whatever from all of these parts of the world. But what do you think you will be keeping up to date with the databases, for example, like adding in the years to come, for example, huge gaps in South America or the US, Africa, for example, or huge Asia?
Yeah, no, that'd be super interesting. I mean, by the time I started working on this was the beginning of my PhD. I was just like struggling to understand what I was doing. So I remember like we gathered this collection of samples, and this was 2 years ago. But now also the Ancient Metagenome Directory has a lot more samples, which of course are going to make the whole microbial source tracking problem much more interesting. So yes, that would be interesting. So to give an update of that matrix of sources and sort of run it with on our own with our own resources, and then spare you guys the time of building that. Yeah, which is also much more inclusive, because it's true. I mean, there's nothing in the south of America or Africa. Yeah.
Any other question?
I have another one about the akmerBroom then. So for the reads that come from really highly conserved regions of the genome, how do those end up being treated? Is there a way that you can distinguish whether those are genuinely from an ancient sample or an ancient organism versus modern contamination?
From the reads that come from a no, we don't do any sort of pre-processing based on how conserved the reads are. But I guess there's something you could do. That's something you could do somehow. If you already know which regions those are, then you could like pre-select those. But you're talking about like your input sample or for the k-mers that we use to build the Bloom filter?
Yeah, for your input samples.
For your input sample. So I guess, I mean, one thing I'm thinking about is like if you already know which regions are preserved, and if they're going to, if you have this hypothesis that that's going to give a differential result between the regions that are preserved and are not, then you could like split them and test the method on both, and then see what the difference is. But no, that's not something we have implemented yet. No.
Any other question? Last chance. Fine. I think, yes, if there is no more questions, I thank you, Camila, for accepting my invitation for this seminar. I think it was great. So hope, yes, one thing I wanted to say is, I'm sorry, I recorded the meeting, so Camila agreed. But so I'm going to stop the recording right now, and I will